I'm going to talk about the cell cycle and its control. The cell cycle starts with G1, which stands for first gap phase. The cell spends a majority of its time in G1 phase, where it just grows and develops. From G1 phase, it can do a couple of different things. It can enter G0 phase, where it doesn't go through mitosis and it just does normal functions. Or it can enter apoptosis, which is programmed cell death. Or once it passes the G1 checkpoint, which checks to make sure the cell is ready to divide, it goes into the S phase, which stands for synthesis. During synthesis phase, the DNA is replicated. The S checkpoint makes sure that there are no errors in the DNA. After that, it moves on to G2, which is the second gap phase. In the second gap phase, it double checks that there are no errors in the DNA and that all the chromatin are connected to microtubules. After it has passed the G2 checkpoint, it moves on to M phase, which is mitosis, where the cell actually divides and the two daughter cells enter back into G1 phase. There are many different things that regulate the cell cycle, and the main ones occur at the checkpoints throughout the cycle. The proteins that control the procession through the cell cycle are called regulators. First off, we have inhibitory regulators, which slow the procession through the cell cycle or inhibit it. These are coded by something called tumor suppressor genes on the DNA. Some examples are the RB protein, P53, and P27. The main problem with inhibitory regulators occurs when there's not enough of them and the cell is allowed to move through the cycle when it's not supposed to. We also have stimulating regulators, which, as the name suggests, stimulate the cell and speed its procession through the cycle. They are coded by a type of gene called oncogenes on the DNA. When a mutation occurs in oncogenes, they become proto-oncogenes. Proto-oncogenes cause an increase in the production of stimulating regulators, which can cause the cell to become cancerous and divide too frequently. This often causes a buildup of cancerous cells we call a tumor. Some examples of stimulating regulators are MYC and cyclin, and MYC actually upregulates or increases the production of cyclin. Cyclin is a very important regulator that is going to be the focus of the second part of this video. The stimulating regulator cyclin is an important protein that drives the cell cycle. So here we have a cell in G1 phase, and there are a lot of different things going on here, but we're just going to focus on the important ones. So these are proteins called cyclin-dependent kinases, or CDKs, and this is going to be cyclin. So throughout the cell cycle, cyclin is produced and its concentration increases in the cell. So it just floats in the cytoplasm until it finds a CDK and it binds with it to form a cyclin CDK complex, which is this thing. So cyclin is being produced and bound to the CDK, and cyclin CDK complexes phosphorylate a variety of proteins to either activate or deactivate them in order to push the cell through the cell cycle. And we have all our cyclin CDK complexes. Once we have enough of them, the cell is propelled into the next phase due to the activity of the proteins that were activated or deactivated. And the cyclin is actually used up and destroyed through this process, and it starts over again, which is why the cyclin concentration fluctuates throughout the cell cycle, while the CDK concentration does not, because it is not destroyed, it's a protein. And the process repeats throughout the S phase, and like all the phases. And once we have all of our cyclin CDK complexes, is again pushed to the G2 phase. And in G2 phase, the cyclin CDK complexes actually have a special name. They're called maturation promoting factors, or MPFs, and they initiate mitosis. So once the process has been repeated, it goes into M phase, where the cell divides. And there are many different types of cyclin, and each phase uses a different type so that the cell knows what phase it's in and what it's supposed to be doing.